to learn about cell organisation and enzymes. So first of all, we know that we have cells and cells can be specialised. Cells become specialised through differentiation, and specialised cells will carry out particular functions in the body. So our cells will come together and are then organised into tissues. So similar cells get organised into tissues. And the tissues are the group of cells working together for a particular function. So for example, if we have muscular tissues, they contract, which means that they shorten, and that will therefore move whatever the muscular tissues are attached to. Glandular tissues will make chemicals such as enzymes and hormones, and they'll be secreted. And then epithelial tissues will cover parts of the body, and epithelial tissues can be ciliated if they come from ciliated epithelial cells, or they may not, depending on which part of the body they are covering. Then the tissues will then be organised into organs. So for example, we have the stomach, and the stomach is a different group of tissues that are working together for that particular function. So for example, in the stomach we will have muscular tissues that need to contract to churn up the food. We'll have glandular tissues which make digestive juices to help digest the food that's inside the stomach. And then we have the epithelial tissue, which covers the outside of the stomach. And for the stomach, the epithelial tissue is not ciliated. Then the organs will be organised into organ systems. And our example here is the organs coming together in the digestive system. So in the digestive system, the organs which we have are, we have the salivary glands here, and they produce some digestive juices. We have the stomach, so we go down here into the stomach, and the stomach and the small intestines, which is this inside part here, will digest the food. Now the liver here, so this part here, which is the liver will excrete bile into the juices just before they go into the small intestines. And what the bile does is it neutralises the acidity, because we don't want the acidity to damage the small intestines there. And then we have the large intestines, and the large intestines will absorb water from undigested food, which then leaves the faeces, which is then excreted. For our bodies to function properly, we need these special chemicals called enzymes. Now, enzymes are biological catalysts. You would increase the speed by increasing the temperature. Now the issue with doing that inside living things is that if we increase the temperature and we increase the rate of reactions within living things in that way, that would also increase the rate of reactions that we don't want going on. So that's really not a good way to increase the reaction of the actual things that we want to happen. So what happens is we use these enzymes, which are the biological catalysts, to speed up the reactions, but only of the ones that we want to happen. So enzymes reduce the need for higher temperatures, for increasing temperatures. Enzymes are large proteins. So that's what enzymes are. They are large proteins. And they are made up of chains of amino acids. But different enzymes have different unique shapes which make them that particular enzyme. So if we're considering enzymes, what we have to do is we have to recognise that enzymes have a particular shape. So if this is my enzyme here will have this particular shape here, which is called the active site. And what that means is that the active site will only fit into it a particular substance. So a particular substrate will only fit into this enzyme here. If it's a different shape, it will not fit in, so this enzyme would not work for it. So if I have a substrate that has the correct shape, then this enzyme would work on this particular substrate. So the enzyme would have the substrate fit into it like this, because we've got the matching substrate fitting onto the active site. And this is called the lock and key method. And when this happens, the enzyme will then start working and it will break down the substrate, which is then released. So the substrate gets broken down and is released. Enzymes will only work at particular temperatures. So if we consider a graph where we have temperature in degrees Celsius against the rate of reaction with the enzyme. To start with, as you increase the temperature, the rate of reaction will increase up to the optimum temperature. And the optimum temperature for most living things, we would be looking at round about 37 to 40 degrees Celsius. After that particular temperature, the temperature is too hot. So what that means is the enzyme becomes denatured. So the rate of reaction increases up to the optimum temperature, and then because the enzyme becomes denatured, the rate of reaction will decrease dramatically. We can also look at the pH if we are looking to increase the rate of reaction. So again, rate of reaction here. Now, the pH, if it's too high or too low, the enzyme won't work. The enzyme will only work at a particular pH. So, for this particular enzyme, if the pH is too high, the enzyme is not going to work. It's become denatured. Now, the optimum are usually pH 7, but not always. So this is the optimum. So for example, as before, when we looked at the stomach is acidic. So it has hydrochloric acid in there, which is churning up and digesting the food. Now, hydrochloric acid, with it being an acid, has a low pH. The pH for the enzymes that are in the stomach would probably be a lot lower for their optimum pH. So they would be probably working at a pH of 2 or 3. So now we're going to consider some different enzymes used to break down larger molecules within our bodies. So first of all, if we consider our larger molecules, and the first one that we are going to consider would be starch, which is also a carbohydrate. The enzyme which is used to break down starch or the carbohydrate could be amylase, and amylase is an example of the enzyme carbohydrase. Okay, and once the carbohydrase does its work on the starch or the carbohydrate, the smaller molecule that is produced would then be a sugar. For example, it could be something like maltose, or it could be dextrin. And the carbohydrate can be made in the salivary glands. It can be made in the pancreas, and it can be made in the small intestines. Now the next molecule we will consider will be protein, and quite often we need to break down proteins. So we'll put our protein in here, and the enzyme used to break down the proteins is actually called protease. Okay, so when protease breaks down the proteins, then we have amino acids produced. So amino acids can come together to make proteins, but proteins, when they're broken down with the use of the enzyme protease, we get amino acids. And the protease can be made in the stomach, it can be made in the pancreas, and it can be made in the small intestines. The third large molecule that we will consider now will then be lipids. And lipids are just fats or oils. And I always remember that, because if you think of liposuction, you're thinking of having fat removed. So lipids, liposuction, so lipids are fats or oils. Now, the actual enzyme that breaks down lipids is called lipase. And lipase will break down the lipids into glycerol and fatty acids. And lipase is produced in the pancreas as well as the small intestines. Now for your GCSEs, if you have to remember specific places where these particular enzymes are produced, I would recommend remembering the pancreas and the small intestines, because all of these different enzymes that you have to know about are all produced in the pancreas and the small intestines. You can see that here. So carbohydrase breaks down carbohydrates or starch, which is a form of carbohydrate. Protease breaks down proteins, and lipase breaks down lipids. Okay, now let's have a go at some questions. You may wish to pause and have a go yourselves before we go through the answers. So the first question is, we've got to label parts A, B, C and D on the digestive system. If we look at part A, that part is within the mouth and that part is the salivary glands. If we look at part B is where bile is produced and that is the liver. Then part C is where food is churned up and we also have hydrochloric acid in there, and that is the stomach. And then part D is where we have enzymes such as lipase and protease and amylase being produced, and that is the pancreas. Now, if we have a look at our next question here, we're going to be looking first of all at the graph with enzyme X and enzyme Y. And the first question is, what's the optimum pH for enzyme X? So the pH is on the x-axis, the rate of enzyme activity is on the y-axis, so we need to extrapolate our graphs downwards to work out the pH. So for enzyme X, we find out where the highest point is, and you would draw a line downwards, and if you drew a line downwards, that would come to pH 2. We would then, for the second question, do the same for enzyme Y. You would get your ruler and draw a line downwards, and if you drew a line downwards, it would be halfway between pH 7 and 8, which gives us a pH of 7.5 for the optimum pH. Now, which one of these enzymes would be found in the stomach? Now, the stomach has a very, very low pH, and it's usually pH around about pH 2. So when we looked at our enzymes, the one that's working at pH 2 would be this one here, which would be our enzyme X. Now, if we look at the next question is asking us, where in the digestive system might the two protease enzymes be found? So first of all, if we look at our enzyme X, we know that enzyme X is a very low pH, which we have already identified as being in the stomach. So the only one of these answers going downwards where we have the stomach is this last one here. So then we look across to enzyme Y, which had an optimum pH of 7.5. So would an enzyme that has an optimum pH of 7.5 be produced in the small intestines? Yes, it would. So therefore, our answer is this last answer here. And that brings us to the end of this session. If you like this, please subscribe and like, and I hope to see you in some more sessions.